Good afternoon. What we're studying here now is cell parts. We got that test on Friday. One of the first cell parts that we studied was the cell membrane. And now we're moving on to another organelle known as the endoplasmic reticulum. Endoplasmic reticulum, when I take apart the name itself, endo inside, plasmic jelly-like fluid, and reticulum is a network. A net type, and that's exactly what this organelle does. It is in the cytoplasm between the nucleus and the cell membrane, and it provides a network really for molecule assembly. The molecules, of course, are in the cytoplasm, and as they are needed to make the additional organelles or to repair organelles or to conjure up those chemical reactions that produce those products that are needed, this is where it occurs. It's a series of membranes. When I think about this, I think of a piece of folded tissue paper back and forth. It provides for a surface area, very thin. It's also almost an assembly line as it wants to draw those molecules in for the chemical reactions to produce products for the living cell. There are two types. The first type is the rough ER. Those are the ones that will be dotted with the ribosomes. And the smooth ER, again, it's the membrane but no ribosomes. And like I said before, it's a molecule assembly point to make protein. This is a photograph, kind of a cartoon drawing of the endoplasmic reticulum, looking deep into the cell's interior. Here would be your nucleus. I would identify that as genetic material. That would be a nuclear pore here. But this series of folded, almost looking like tissue paper back and forth, would be your endoplasmic reticulum. Notice that this is dotted with ribosomes. They're the small microscopic bodies that go on. It almost looks like maybe that outside one out here doesn't have the microscopic bodies. Again, that would be the smooth ER. So the difference between the rough and the smooth: presence of ribosomes, no ribosomes. Here I have another picture of the endoplasmic reticulum, just from a different point of view here. We've got our cells, the nucleus, and they're calling that the nuclear envelope. You know, we could call that the nuclear membrane if we wanted to. Nuclear pore, those will be your entrances and exits in and out of the nucleus, depending upon which molecule is allowed in or allowed out. But then really close nearby, you have got this series of folded membranes, the endoplasmic reticulum, and they're making a difference here. This one is studded with ribosomes, therefore it's going to be rough. This over here is smooth, no ribosomes as well. Again, assembly of proteins here. Those proteins will promote the living activities of the cell. Another organelle found in the cytoplasm of the cell would be the Golgi bodies. You may see them called Golgi apparatus. Don't let that confuse you. It's not ambiguous. Just two names for it. Like it says, a series of flattened sacs, and their job is to process and package proteins and lipids for expulsion from the cell and makes secretory vesicles. So you'll find these guys more numerous in cells that have something to produce, that have I'll call it a chemical or a substance. It's, you know, it's going to be a living matter. You know, kind of like, you know, when your nose runs, you know, there's cells lining the nasal cavities, and they're producing that mucus at the right time, and your nose is running. And otherwise, they still produce some mucus, but you really know when there's a cold. How about insulin? You know, another thing coming out of your pancreas. How about your liver? Oh my gosh, you know, you got lots of substances that are made that are highly specialized. Now, a secretory vesicle, you see in secretory secrete, a vesicle is just a container, so it's actually making items, making substances, it's packaging them, and they're leaving the cell. They're leaving the cell to go do their business elsewhere. And after that we got lysosomes, and they're kind of tied into the Golgi bodies because they will make a package, and it's retained for the cell's cytoplasm, so they don't leave the cell, they stay in the cell, but they're enzymes that are in the package. Remember, enzymes are proteins, don't let that get you, that digestion within the cell. So they are working in the cytoplasm to go ahead and digest large particles. Maybe they're cleaning up debris inside the cell from some cell part that worn out. They want to clean it up and break it back down to its simplest molecules and, recycle what molecules they can. And they're affectionately known as suicide bags because of this capability. Here is a real nice diagram image, got it off Google Images. We're working again with the lysosomes. As you can see, we've got ourselves the rough ER making the proteins. It's a transport vesicle. It's going to the Golgi body, and the Golgi body is producing the lysosome. And it appears like the lysosome is actually looking for a particle inside the cytoplasm. It's not one that leaves. Inside the cytoplasm. Here, nice diagram, lysosome engulfing damaged organelles. So it wants to recycle the molecules of the organelle that it can. So it's got to break it down. And this is kind of like the garbage disposal in a way, but breaks it down to a point where some of the parts can be recycled. This is happening inside the cytoplasm of the cell. Remember that the Golgi apparatus is also responsible for making secretory vesicles. I don't see one. Do you see one here? I don't see one. Secretory vesicle that would actually come out here and leave the cell. So you've got the two that are going on there. All right. Thank you.